because of this hydrogen atom. This RNA molecule is more delicate, or it is less stable than DNA because of the presence of this unreacted OH group at carbon number 2. This one, it can be attacked by damaging molecules. So this one, it makes RNA less stable than DNA molecule. DNA, it can be stable for a long period of time. It can be detected in fossil, which is length, which is buried for a long period of time, or we can detect DNA molecule from okay. Is it not audible? Do you hear me now? Hello? Okay, some of you, maybe you are not using proper. Thank you. Okay, most of you are hearing me, so I will continue. Thank you. Okay, DNA, it contains deoxyribose sugar. It is deoxy form. It lacks oxygen at carbon number 2. This one contains hydroxyl group at carbon number 2, and this makes the RNA molecule less stable than DNA molecule because of unreacted OH group. This one, it is involved in the phosphodiester linkage. Is in the linkage. The carbon number OH group is also involved in this phosphate group. Carbon number 1 OH group is involved with in the base attachment. So we have unreacted OH group. This will be attacked by these other chemical agents. So this one becomes less. There are 3 types of RNA molecule. All of them, they are important for making protein. They have a role in protein synthesis, but their function is different. For example, the messenger RNA is formed as a result of DNA transcription. When the DNA is expressed, it will copy the information into messenger RNA. Not the whole linkage, but segment of DNA, it will be transcribed into messenger RNA, segment of DNA. If we have 2 meter length DNA, only segment of DNA will be transcribed into messenger RNA. Our DNA is double-stranded, you know this one. So from these 2 strands, one of the strand, it will be copied into messenger RNA. One of the strand only, it will be copied into messenger RNA. Then this will be used for protein synthesis in the cytoplasm. The second type of RNA molecule is transfer RNA. This one is used to bind amino acid in the cytoplasm and then collect them into toward this protein synthesis location. Transfer amino acid to the site of protein synthesis by this transfer RNA. Ribosomal RNA, as I said, it is used to make ribosome. So ribosome, it is our machinery to synthesize the protein molecule. Ribosomal RNA, it is important to make ribosome. Then ribosome is important for protein synthesis. These 3 types of RNA molecule, they have different size, function, and stability. The dominant type of RNA molecule is ribosomal RNA, and this one is transfer RNA is the second most dominant type of RNA molecule found in the body or in our cell. Messenger RNA is accounting less percentage. The lengths, based on their lengths, they are differ. For example, this one is the largest in lengths, even if it is accounting the smallest percent of the total RNA molecule. This one is the longest type of RNA molecule, and the shortest one is it is transfer RNA. The messenger RNA, this one accounts only 5% of the total cellular RNA molecule, which is the smallest in percentage. This messenger RNA, after transcription, we have transcription reaction. Transcription, this will be modified into a type of messenger RNA. For example, we have DNA this DNA will be transcribed into messenger RNA, which is immature or pre-messenger RNA, we call it. Pre-messenger RNA will be formed. The pre-messenger RNA will be modified. There is post-transcriptional modification. Messenger RNA, this will be modified into matured type of messenger RNA. How it is modified? It is modified by adding Brook. Okay, the pre-messenger RNA will be modified by this. There are three important processes. The first one is capping at 5' end of this messenger RNA. There is capping and poly-A adenylation at 3' end of this messenger RNA. The third modification is removal of introns. We remove introns. Removal of introns. It is the modification in the pre-messenger RNA. As a result of this, we have matured first methylate 7-methylated cap the 5' end of messenger RNA. Poly-A adenylation is addition of a number of adenine at 3' end of messenger RNA. These are they are modified because this messenger RNA is delicate molecule, or it is less stable. It can be degraded by exonuclease enzyme. So to have this protection, we have capping at 5' end and 3'A polyadenylation occur because of this exon. So the enzyme they can before translation process. Plus the important initiation synthesis, initiation of protein synthesis. Cap or ribosome recognize 5' capping. As a result, there is initiation. Tell me the reason. There is no post-transcriptional modification in the prokaryotic cell because they don't have. There is no nuclear. Immediately after transcription, there is a translation process, or sometimes there is simultaneous transcription and translation. Transcription. Since you are okay, this one, it is messenger RNA. It is formed by this transcription of DNA molecule into messenger. It will be protein. There is so this is transcription process, and this one is translation process. Transcription means copying of nucleotide DNA information into messenger RNA. They use the same language. Both of them, they are synthesized by nucleotides, or they use the DNA to messenger RNA. Then after messenger RNA synthesis, this will be used for protein synthesis by translation. Translation means conversion of one language to another language. Messenger RNA, it is nucleotide language. Proteins, they are amino acid language. They are made by amino acids. Therefore, they use a different language. So to have this, we have translation process. Translation is occur in the cytoplasm. So in the prokaryotic cell, there is no nuclear membrane. The nucleoid or their DNA is found in the cytoplasm. Therefore, there is simultaneous transcription and translation. They have a simultaneous transcription and translation process before modification. But in our case, in the human body or in the eukaryotic cell, there is post-transcriptional modification by capping at 5' end and polyadenylation at 3' end, plus removal of introns. Introns they are non-coding regions. This DNA to have two regions, coding and non-coding region. Coding region, it is called exons. We call it exon. Exons, they are a coding region. They have information for protein synthesis. Exons are coding region. Okay. This one is a coding region. Introns, they are non-coding regions. They are junk material found in the DNA molecule. This one is for the protection of DNA, DNA protection. Non-coding region. This one is non-coding. After transcription, there is removal of introns by splicing out. Spliceosome enzyme remove introns. So this will be the third modification. The pol A wallets, they contain about 200 successive adenylate or adenine residue. This one is important for protection of messenger RNA from exonuclease attack, plus to transport messenger RNA from nucleus to cytoplasm. There is transport of messenger RNA into cytoplasm. It is occurred by this pol A adenylation at 3' end of messenger RNA. Messenger RNA, it consists about 1,000 to 10,000 nucleotide sequence. It is the longest in length. This one is the size of messenger RNA. It can length from 1,000 to 10,000 nucleotide length. This messenger RNA, it has a different life span. It can live for few minutes to days. Most of the time, it is degraded after giving this translation process. Translating messenger RNA into protein. After this function, there is a degradation or removal of messenger RNA to avoid excess protein synthesis. Okay. So this is lifespan of messenger RNA. It can length from few minutes to days. Most of the time, we have less than minutes of lifespan for this messenger RNA because of avoiding excess protein synthesis, or as a result of messenger RNA, if this one is functional for more time, it will be transcribed or translated more than one times. In prokaryotic cell, the 5' end of messenger RNA sequence contain AG-rich region. There is adenine guanine rich region in the 5' end of messenger RNA. This one, it is we call it Scheindahl-Garneau sequence, and this one is important to have initiation of protein synthesis. It will be used for the attachment of ribosome with messenger RNA. As a result, there is initiation of protein synthesis. This is for prokaryotic cell. But in the case of the eukaryotic cell, we use Scheindahl-Garneau sequence. This one is a consensus sequence found from 5' end of messenger RNA. It will be used for protein synthesis. Okay, some prokaryotic messenger RNA has secondary structure. There is self-folding and intrastrand base pairing formation. The complement bases, they are base paired for the formation of secondary structure. They form a secondary structure by intrastrand base pair formation. As a result of this, there is hairpin formation or loop-like structure formation. For example, assume this is one strand, then it will be folded and they form intra-strand base pair formation. This one, it is secondary structure. Like this, it will be formed hairpin or loop-like structure. It is formed in some of prokaryotic messenger RNA. They form a secondary level of nucleotide structure. The second type of RNA molecule is transfer RNA. This one, it accounts 15% of the total cellular RNA. It is the smallest in size. It contains 50 to 100 nucleotides. This transfer RNA function, the function of this transfer RNA is to collect amino acid to the site of protein synthesis. There are 4 arms for this function. For each transfer RNA, there are 4 arms: amino acid binding arm, dihydrouridine arm. This one is for binding the active site of amino acid transfer RNA synthetase. This enzyme is for activation of transfer RNA. The active form of transfer RNA is amino acid binded form. It is amino acid binded form. So this enzyme, it is bind with dihydrouridine or dihydrouracil. This one will be used for binding the active site of the amino acid transfer RNA synthase enzyme. The third arm is 𝒕𝒖𝒓𝒄𝒕. This one is 𝒕𝒖𝒓𝒄𝒕 or thiamine and pseudouridine. This one is pseudouridine and cytidine. They are found on the third arm. This will be used for binding of ribosome. Bind ribosome, and there is a protein synthesis by this ribosome. By binding this ribosome, there is a protein synthesis. So this one is psi. We call it psi. It is found in the third arm of transfer RNA. The fourth arm, it is anticodon arm. This one to read the information in the messenger RNA, then transfer amino acid accordingly. For example, this is our messenger RNA. It contains nucleotides. There are nucleotides here. Adenine, thymine, guanine, cytosine. They are found on this messenger RNA. This will be read or bind with this anticodon. These are codons that are found in 1 liter represent one nitrogen base or one nucleotide. For example, this one is one codon. It will be attached by the codon arm of transfer RNA. Assume this is one codon and transfer RNA. It is look like this structure. There is such kind of structure. Okay, this one is transfer RNA. This one is 5' end of messenger transfer RNA. This one is 3' end. At 3' end, there are three nucleotides, consensus nucleotide, adenine, cytosine, bind amino acid. This one is amino acid binding arm. Amino acids, they are carried by transfer RNA at 3' end. This one is 3' end. Okay, this one is DHU arm. DHU arm or dihydrouridine arm bind the active site of enzyme. As a result, this transfer RNA will be activated into amino acid transfer RNA. No, amino acid transfer RNA. It is active form of transfer RNA. This one is anticodon arm. There is base pair formation between the codon region of messenger RNA and anticodon region of this transfer RNA. There are three nucleotide attachments here. There are three arms here. One of these nucleotide attached is the first letter. The second will be attached to this one. For example, if we have adenine here, there is thymine here. Is it correct, thymine or uracil? It is paired with adenine here, and if there is cytosine here, there is guanine here. So UAC, it is anticodon found on this transfer RNA, bind specifically to this codon region of messenger RNA and transfer amino acid to this protein chain. There is proteins here, for example, like this. There are proteins. So this amino acid, it will be transferred to this peptide chain. This is a peptide chain, and then this amino acid, it will be transferred to this peptide chain by using this codon. Every amino acid, they have their own codons. They have their own codons. For example, we have 20 amino acids. For this 20 amino acid, there should be at least one codon. There should be at least one codon, but amino acids, they have more than one codon. Number of codons are 64. There are 64 codons. These 64 codons, they will be distributed for this 20 amino acid. One amino acid can have more than one codon, but one codon is specifically code for one amino acid. Specifically code for one amino acid. There is no overlapping between codons and amino acid transferring. Okay, we'll see later on your molecular biology or neoplasia class or module, number of codon, how 64 codons are formed for this 20 amino acid የሚለውን በኋላ neoplasia class ላይ በደንብ የምትማሩት ይሆናል። እሺ the amino acid arm, amino acid binding arm is it is for amino acid binding or attachment to the 3' OH group of this transfer RNA. The 3' end of transfer RNA contain these three bases, adenine, cytosine, and cytosine. They are found at 3' end and bind for amino acid and transfer to this protein synthesis site. The anticodon arm, this one, it is important to recognize the codon region on messenger RNA. recognize the codon region on messenger RNA. As a result, transfer amino acid to this peptide chain. 5' end of transfer RNA is phosphorylated, and the residue is guanosine. There is phosphorylated at 5' end. That means it is capped capped at 5' end of the chair of this one or psi. There are unusual bases here. Unusual bases, they are found in this transfer RNA. Unusual base means they are modified. For example, uracil is modified into dihydrouridine, plus pseudouridine is also found here, like this psi. They are unusual bases. They are found in this RNA molecule. So this one is used to bind the active site of enzyme, and this one is used to bind the codon region of messenger RNA. Messenger RNA is here bind with ribosome. The large subunit of ribosome, it is used to bind this messenger RNA, and the codons are read by this anti-codon arm of transfer RNA. Extra arm is also here. We have extra arm, which is variable between transfer RNA. TCIC arm, this one is used to bind ribosome, and then there is a shifting this one, and this one is used to bind amino acid. Amino acid, it is binded on the 3' end of transfer RNA. So this is transfer RNA. For the 20 amino acids, again, we have 20 transfer RNA. Each of the 20 amino acids, they are carried by their own transfer RNA. This one, for example, it is for tyrosine amino acid. If you say this one is for tyrosine amino acid, other amino acid, they use a different transfer RNA, or they are carried by their own transfer RNA. The codons here found on the messenger RNA, they are coding specific amino acids. There are 64 codons. Okay, 64 codons are found in the messenger RNA. Out of this, there are stop codons either, or they code for specific amino acids. Stop codons for the termination of protein synthesis, we have stop codons. Okay, the third type of RNA, it is ribosomal RNA. Ribosomal RNA, it is used to make ribosome, and the ribosome, it is important for making protein. Protein synthesis is occurred by using this ribosome machinery. This one is highly methylated. There is methylation of ribosomal RNA as compared to the other types of RNA molecule. Ribosomal RNA with ribosomal protein and magnesium, they constitute this ribosome. They make this ribosome. Again, contain 2 subunits, one big subunit and the second small subunit are found in the ribosome. Each subunit, they contain 2 subunits. Each ribosome contains 2 subunits, big and small subunits are found. The big subunit is used to bind messenger RNA. We have big subunit like this one and smaller subunits. This is smaller subunits found in the ribosome. So the big subunit, it is used to bind messenger RNA. It is found here. The smallest subunit, or the small subunit of ribosome, it is used to bind transfer RNA. There are 3 sites here. A sites, there are A sites, P sites, and E sites. This one is E site. So P site is for peptide bond formation, P is for peptide bond formation, and this one is for E exits. So A is addition of new, the newly coming transfer RNA is added here. It comes and bind on the A site of small subunit, then transfer amino acid to the peptide chain. After transferring the amino acid, it will be exits from E site. There is exiting from sites on the small subunit. It is RNA 5' end, and this one is 3' end of. There are codons here. They are read by transfer RNA. A site, it is for its peptide bond. It is formed at P site, and E is for exiting of the free transfer RNA. So these are the two subunits of ribosomes. 4% one, 80% of the total cellular RNA it is formed by this RNA. Account 80% the total cellular RNA. Messenger RNA is 5, transfer RNA is percent. 80% of the total cellular RNA is made by ribosomal RNA. In ribosome, ribosomal RNA they are found in combination with protein. They are found in together with protein. We call them ribonucleoproteins. This is another name of ribosome. Length of ribosomal RNA 100 nucleotides. So it is found between the two types of RNA molecule. Messenger RNA is the longest one. Transfer RNA is the shortest one. And ribosomal RNA it is found between of in between of messenger RNA and transfer RNA. It have 100 and 600 nucleotide lengths. Ribosomal RNA differ in cell. That means molecular weight and shape of ribosomal RNA. Weight of ribosomal RNA, we have different types of ribosomal RNA. In prokaryotic cell, we have 23S, 60S stands for Svedberg unit. This one is Svedberg. Segmentation RNA. This one is in molecular weight and shape 5S. And the cell, there are 4 types of ribosomal RNA: 28S, 18S, and 5.8S, 5S. These are the 4 different types of ribosomal RNA found in the eukaryotic organisms. So S stands for Svedberg unit, and it is given based on the molecular weight and shape of ribosome RNA. End of ribosomes. 70S ribosome is for prokaryotic ribosome. But in our case, in the eukaryotic cell, there is 80S. There is 80S ribosome. It have larger and smaller subunits. Larger subunit it is 50S, and the smaller subunit is 30S. Here we have 31 proteins. It is made by 23S ribosome and 5S ribosomal RNA. The smaller subunit it is made by 16S ribosomal RNA. This one it contains 60 S ribosomal RNA plus 40S. 40S. So this one is the larger subunit, and this one is the smaller subunit of the eukaryotic ribosome. Okay, when you see this one, it is not summative. It is not summative. 60 plus 40 is 100, but the total molecular weight of this ribosomal RNA is 80 in the eukaryotic cell. It is not summative. You don't have to add this to get the total molecular weight of this ribosome. It is not summative. 50 plus 30 it is not 70. So they are not summative. This is the structure of 16S ribosomal RNA. It is a complex structure. It is found in secondary structure. Okay, thank you very much. We have finished today's class. This is the end of nucleic acid structure. If you have question, now you can raise your question. You can write your question on chat. Okay, how can I share the slide? If you send me your email address, then I can. Yes, we can. Yes, we can form a single stranded DNA molecule when, for example, stranded DNA molecule, but immediately after some time of replication, there is a double-stranded DNA molecule formation. Okay, I can share. The other question is, what does it mean methylated? Methylated means addition of methyl group. There is CH3 or this one, it is methylated, or addition of methyl group to the molecule, it is called methylation. We call it methylation. ለምሳሌ thiamine, it is formed by the methylation of uracil. Okay, addition of methyl group. The other question is, what RNA molecule are exceptionally not single-stranded? For example, transfer RNA is found in the tertiary level, tertiary structure. 16S ribosomal RNA is found in secondary structure. Self-folding is it is found in the RNA molecule. For example, DNA it is formed by two separate strands. There is a two separate strand in the DNA molecule, but RNA they have a single strand, but this single strand it can be folded on itself. There is self-folding, like this one. We have seen the example for this transfer RNA. I hope can answer your questions. Could you please repeat the part on how ribose can be produced from glucose? Okay. Ribose it is formed in HMP pathway. There is hexose monophosphate pathway or pentose phosphate pathway. Through HMP pathway, we form ribose phosphates. There is a ribose phosphate formation. For example, the glucose 6-phosphates, glucose 6-phosphates, it is the one which is used for glycolysis pathway. This will be converted to pyruvate in the glycolysis pathway, but there is one alternative pathway. The alternative pathway is to convert glucose, a 6-carbon sugar, to 5-carbon sugar. There is ribose phosphate formation. This will be given in the third module, I think. In the third module, it will be given. So ribose phosphate is formed here. Ribose phosphate is formed from this glucose through the HMP pathway. We have a single-stranded DNA, but it is after transcription, immediately after the transcription reaction. Okay, thank you very much. A form of DNA contains 11 nucleotides, so how it is possible how A form DNA shorter? No, A form of DNA contains 10 nucleotides, not 11. The A form, it contains 11 nucleotides. Okay, A form, there are 3 forms of DNA. A form DNA, this one is the most stable one at physiological condition. It contains 10 nucleotides. There are 10 nucleotides per one helical turn. Per one helical turn, we have a 10 nucleotide length. The B form DNA is formed as in alcoholic condition or in the dehydration form. There is a B form. Sorry. This one A form, it is formed during dehydration or in alcoholic form. In alcoholic solution, there is A form nucleotide formation. This one, it contains 11 nucleotides. 11 nucleotides are found in the A form DNA. Okay, this one is shorter in length than B form DNA because per one helical turn we found 11 nucleotide. Even if the number of nucleotide is greater than the B form DNA, but the length is shorter. There is dehydration of the B form DNA into A form DNA. This occurs by dehydration, and the length it becomes shorter as a result of this dehydration process. The Z form DNA is less standard and it contains per one helical turn. This one is very important. What is the use of dihydrouracil loop in the transfer RNA? It is to bind the active site of transfer RNA synthetase enzyme, the enzyme which can activate transfer RNA into amino acid transfer RNA. Free form of transfer RNA is activated into amino acid transfer RNA by this enzyme. So the enzyme, it have active sites where catalysis is occur. So DHU arm, it is used to bind the active site of this arm, and then as a result there is activation of transfer RNA into amino acid transfer RNA. Do bacteria DNA has introns? No, they don't have introns. They are made by only exons region. There is no introns, as there is no modification in the bacteria cell. We don't have capping, we don't have, they don't have this 3' polyadenylation, there is no intron removal in the bacteria. Introns or non-coding regions are not found in the bacteria cell. Okay. The other question, I think I have finished. Actually, I will send this slide for one of you, then you'll share it for the other. For example, Johannes G. Asra, Johannes G. I will share it for this student, and you'll share it. Johannes, are you there? Okay, thank you very much. Good night. Good day.